Good day, everyone. To everyone listening to this class, I'm excited to bring you another topic in introductory microbiology. Today, we'll be looking at an interesting topic called molecular phylogeny markers. Think about it. It is a topic that talks about ancestral lineage of microorganisms, and these markers are what helps us to distinguish them. So, very quickly, we'll be looking at what are these markers, how are they used, and what are they applied, how do we apply them. So molecular phylogeny markers are tools, they are molecular tools that utilize specific molecules as their markers to reconstruct evolutionary relationships between different microorganisms. When I say evolutionary relationships, how they evolve, their ancestral lineage. So these markers consist of genes or sequences within genes. They evolve at a predictable rate and they offer us insight into the lineage and relatedness of microorganisms. So in this lecture, we will be looking at, like I said, different types of markers and their significance in microbial identification. So phylogeny helps us to understand the evolutionary relationship between different microorganisms. The word phylogeny comes from the Greek word phylon, which means tribe, clan, or race. The genesis is the origin or source. So it talks about how organisms are related. So by analyzing this genetic makeup of organisms, particularly their DNA or RNA sequences, we can reconstruct their evolutionary history. Just like physical features can run in families, you can see people have similar eyes, nose, finger, hair. Microorganisms also inherit genes from their ancestors. These genes act as clues in our investigation. So if two microorganisms share a high degree of similarities in specific genes, it suggests they may have descended from a common ancestor. But if they have a very far, high degree of dissimilarities, then we can clarify them to be similar. So this is just the basic thing the basic thing about phylogeny. But we have markers that we use in this various investigation. So phylogeny is the evolutionary history of group of microorganisms, so species and so on, and it focuses on order of branching from a common ancestor, sometimes estimated time point of their divergence. So we use sophisticated tools to compare nucleotide sequence of microorganisms in the order of A, C, T, and G, which we all know represent adenine, cytosine, thymine, and guanine. So in these genes, these sequences are used in comparison. The more similar these sequences are, the closer the evolutionary relationship between them. That means if they both have this sequence in the same order, then it gives us a clue on these similarities to give us an understanding of the fact that they are more closely related. Sometimes mutations or changes in the DNA sequence can accumulate over time. So in these cases, microorganisms with fewer differences in their gene sequences are considered closer relatives. Those without any differences are considered the same. So when we have few differences in their nucleotide sequences, that is the A, C, T, and G, we say if we have a few very few differences, then we say they are close related. But if there is a wide difference, then they are not related, because this is the basic tool for comparison, which are used to distinguish them from one family or the other. Beyond this species differentiation and understanding, phylogeny can help us to trace the history of organisms. It helps us to classify microorganisms on the basis of their evolutionary history. This classification usually helps us to understand their similarities and differences. It helps us to predict their functions, in the sense that if you know a microbe close relationship, close relative, close species, and their functionalities, then we can make educated guesses, hypotheses about the function, the potential function of newly discovered microorganisms. This phylogeny also helps us to trace disease outbreaks beyond species. Like I said, it helps us to trace disease outbreaks by analyzing the genetic makeup of pathogens. When we look at the sequence of a pathogen, phylogeny can help us to track the spread of infectious disease. We can identify the source of an outbreak and understand how the pathogen might be evolving over time, because we can look at the sequence to relate it with known pathogens and so on and so forth. The molecular phylogeny is a field that studies the relationship between organisms or the genes. So in molecular phylogeny, the relationship among organisms or genes are studied by comparing the homologous DNA or protein synthesis. So the homologous DNA or their protein synthesis are what gives us a clue. And whenever there is the differences in their sequences, that is their DNA, it reflects their genetic divergence. It gives us this understanding of how different they are. So in terms of comparison, we use the DNA and protein sequences for molecular phylogeny. So in this approach, we have the traditional phylogeny approach and the modern day classification using phylogeny. For traditional phylogeny approach, microorganisms have been identified using observable characteristics. The traditional phylogeny approach research played crucial role in the understanding of their evolutionary relationship in former times. In those times, this method relied on analyzing various characteristics of microorganisms and group them based on their presumed evolutionary history. Because this approach focused on observable characteristics of microorganisms. When we say observable characteristics, we are talking about their morphology, their shape, their size, presence of certain structure like flagella, capsules, and so on. It also focuses on their physiology, that is, their growth requirements, temperature, oxygen, pH, and other metabolic capabilities. This is used in classifying them into a presumed evolutionary history. Then it also use staining properties, staining techniques like gram staining, which differentiate bacteria based on their cell wall and structure. However, microbial phylogeny has undergone a significant revolution with the advancement of molecular biology techniques. These techniques provide a more accurate and objective way to reconstruct evolutionary relationships and to classify and reclassify and declassify microorganisms compared to traditional approaches based on phenotypic characteristics. So, traditional phylogenic approach rely on physical characters. When we compare both, the, the traditional phylogeny compare based on morphology characteristics to infer their evolutionary relationship between one organism or the other. Whereas in the modern day method, it utilizes the nucleotide sequence of the DNA and RNA, as well as the protein synthesis, which are determined through advanced techniques. So when we compare both methods, we see that the modern day classification based on sophisticated tools and molecular methods helps to give a more accurate evolutionary information of microorganisms. So if you look at the classical versus modern approach, previously the classical method uses morphology features which determine the identity, but currently the modern approach leverages on the power of modern techniques which use the DNA and RNA sequences, also protein amino acid sequences. So in using this, it helps us to now look at their similarities and help us to classify them. So the dissimilarity among the sequence will indicate genetic divergence because of molecular evolution during the course of time. So that means in the course of time, there have been mutations, probably mutations, change in their sequences, which has allowed this organism to evolve in a different characteristic, different nature, differently from their ancestor. So when we now begin to compare, we now begin to see how differently these organisms have evolved over time. And this can only be made possible when we investigate their similarity in their DNA nucleotide sequences. High similarities indicate close relationship. High level of differences indicate they are not related. The same sequences indicate the same organism. So, one of the molecular markers that are used is the 16S ribosomal RNA. 16S ribosomal RNA is an important tool that is used in identifying microorganisms. By sequencing of the 16S RNA gene, it gives us a clear information of organism identity. The 16S RNA gene is a well-established and standardized technique. The sequencing of the 16S RNA gene is a well-established and standardized technique with readily available protocol in many labs and kits which allow for easy comparison of data that are generated from different laboratories and so on. And it provides an extensive large database we call gene bank. In times to come, we'll be looking at what are these gene banks. And this gene bank consists of several 16S rRNA sequences from vast array of organisms. It's a bank of genes, so we call it gene bank, which allows us to deposit sequences of different organisms. So when a new organism is being compared, it allows for comparison of a newly sequenced microbial 16S rRNA to a known sequences in the gene bank, which helps in identification and aligning the sequence, which is the ACTG, with the known one. If we now sequence a new microorganism, by comparing its sequence with the previous ones in the gene bank, we will be able to now check if they have similar sequences or dissimilar sequences. This will give us an information of how related they are. Now this explanation is better observed when we use molecular tools. But because this is a basic class, we cannot go deeper into this. As the class advances, we'll be looking at these tools that are being used and how these tools are being carried out to effectively determine how alignment are done. However, because this is a basic class, we just want to introduce what is called 16S ribosomal RNA as a marker. And why is this 16S used? It is used for several reasons, because of the fact that it is very efficient. It is universal. That is, the 16S rRNA is present in all microorganisms. It is found in all living organisms, from bacteria to archaea to plant and animals. So this universality makes it a powerful tool for comparing organisms across all domains of life. And it is highly conserved. When I say highly conserved, the 16S RNA has some variability, certain regions that are highly conserved, that is an unchanging portion across all organisms. And these conserved regions allow for reliable alignment and comparison of sequences. The conserved regions are useful for identifying broad taxonomic groups, and it helps us to differentiate between closely related organisms and non-related organisms. So the 16S rRNA gene is present in multiple copies per microbial cell, making it easier to amplify using what we call polymerase chain reaction from even small sample with limited microorganism cell. It can amplify this region and help us in identifying the organisms. So in essence, one good molecular marker that we look at is 16S RNA, 16S rRNA gene. And this is a tree that indicates my phylogeny. It shows how organisms are related. For instance, if you look at these roots here, from these roots we have an extension of another family, ancestral line, which give birth to a branch of this particular taxon, then another taxon. These two bacteria now are different, but they have been traced to this root, and this is the original root that gave birth to this family. And if you look at this, we see another branch which gives back to this bacteria S, and it has some relationship, far different relationship between bacteria 4 and bacteria 3. So as mutation occurs, as change in sequencing occurs, extension in the branches of taxonomy of organisms are discovered, and they begin to have different sequences and different classification. But if you look at their ancestral, because we can have a way of detecting how these organisms are related by this branching, and this is what the 16S help us to do, because there are regions where they have similarities, and there are regions where we can trace their roots from, and this is a tree of phylogenetic tree of microorganisms. So I said previously that the 16S ribosomal RNA is useful because it has hyperavailable regions. It has a conserved region in all microorganisms, and this region act like a fingerprint which allow scientists to distinguish closely related organisms. Then it is relatively stable in every organism. It is a stable protein which is critical for making which is critical for molecular identification of microorganisms. Then its amplification is also with ease. It is easy to amplify this region using molecular methods. So the 16S ribosomal RNA in every microorganism is a stable and hypervariable region with ease of amplification. That is why it's being chosen mostly for bacteria, mostly for bacteria, because bacteria are more in terms of pathogens, in terms of proliferation, they are more ubiquitous. So these regions are used in identifying all these bacteria. Then we have another one that's called internal transcript spacer, call it the ITS. The internal transcript spacer region of the ribosomal DNA. It is a popular molecular marker which is used in microbial phylogeny. So this ITS is a segment of DNA which is located within the ribosomal RNA gene clusters in eukaryotic. This includes fungi, plants, and some protists. The ITS holds particular importance in microbial identification due to its own unique characteristics. The ITS region refers to a non-coding DNA segment which is situated between two genes encoding the ribosomal RNA in the chromosome of an organism. It resides between the 18S and the 28S rRNA genes in the ribosomal DNA operon, which is present in virtually all living organisms, including microbes. So when we say the non-coding region, unlike the RNA gene themselves, the ITS region does not code for proteins. However, it plays a crucial role in the processing of ribosomal RNA molecules during ribosome assembly. It is highly variable. It exhibits significant variation in sequence within and between species. So this variability is particularly pronounced in the ITS region compared to, ITS region 1, which is ITS 1, intertranscribe spacer 1, compared to intertranscribe spacer 2. So we have ITS 1 and ITS 2, and this variability is more pronounced between them. It is considered a non-functional spacer, and like I said, it does not code for any protein, and it does not involve in protein synthesis. The ITS region is flanked by highly conserved sequence on either side, corresponding to 18S, 5.8S RNA genes. So these conserved sequences act as primers. Primers are short oligonucleotides. So they act as primer binding site for PCR amplification, thereby making ITS a convenient target for molecular analysis. So ITS is a good marker for eukaryotic organisms. It is applied for direct function because it is a valuable tool in molecular biology. It is used in species identification and phylogenetic studies, particularly among eukaryotic, because it exhibits a high degree of variation compared to highly conserved ribosomal RNA genes themselves. So it is used for species identification. One of the limitations of 16S RNA is that it may not be accurate for species identification, whereas the ITS application does more in species identification. So if you look at it now, the 16S rRNA can give us the organisms, but when you now go down to the species, ITS applications are more preferable. So the ITS region is also designated, it has a designated barcode region for some organisms which allow for standardized DNA sequence based on identification. The barcoding, genetic barcoding is a region which is designated for identification for some organisms. It allows these organisms to be identified easily because of the barcoding. So advantage of using ITS region is the fact that it is universally present in most eukaryotes and making it applicable across a wide range of organisms. It has a relatively large size which provides good amount of sequence data for analysis compared to some other markers. And it is also flanked by conserved regions of rRNA gene on either side, which allow for design of universal primer for application of the ITS region. When we say universal primer, these are oligonucleotides that can amplify this region. Short oligonucleotides that can help us to identify universally all the organisms, but it will not be able to narrow down on the species. It will give us the amplification of the organisms. Then later on, we can now narrow down on the species. We have other markers which are called housekeeping genes. These are genes involved in essential cellular functions and they are present in all organisms. Examples include gene encoding for elongation factor, RNA polymerase subunit, and DNase gyrase subunit, and so on. So these housekeeping genes can be useful for deeper phylogenetic analysis within specific groups. At this level of introductory microbiology, we may not go deep into this. It's just an information because they are for deeper phylogenetic analysis, such as well as multilocus sequence analysis, which involve sequencing of multiple housekeeping genes and concatenating the sequence for analysis. So the multilocus sequence analysis provides a more robust phylogenetic signal compared to a single gene marker of 16S RNA and can help differentiate closely related strains. But we are not going to look at it. At your advanced class, we'll be looking at all these other markers. But at your basic class, we restrict ourselves to the basic ones, which are known as the molecular phylogenetic markers. So that is for this class, and I hope that you will be able to understand it if you read more. If you have any question, you can drop it at the comment section. I will gladly reply. Thank you for listening. Have a wonderful day.